Hi, my name is Manish. I am a PhD student in Chemical Origins of Life Lab at ISER Pune, situated in India. Today, I am going to talk about the spontaneous emergence of membrane-forming protoamphiphiles from a lipid-amino acid mixture under wet-dry cycles. This talk is primarily based on the research work that I have done so far in my PhD tenure. Modern cell membrane is a complex structure that primarily contains proteins and lipids. This complexity might have been an evolutionary outcome of a dynamic interplay between proteins and lipids, or membranes so to speak, right from the beginning of life. The origin of cellular life on Earth is thought to have followed a fundamental step of protocell formation. Protocell is a cell-like entity that contains both informational and functional molecules which are encapsulated in a membrane compartment. There are two processes which are thought to have been crucial for the formation of a protocell. These processes are membrane assembly from simple single-chain amphiphiles like fatty acids and their derivatives, and a non-enzymatic oligomerization, which could be nucleic acid synthesis from mononucleotides or peptide synthesis from amino acids. Given that both membrane assembly and non-enzymatic oligomerization processes would have simultaneously occurred during a protocell formation in a common milieu, it is imperative to ask how these two processes might have affected each other. There are several studies which have looked at the effect of lipids as a co-solute on non-enzymatic RNA formation under wet-dry cycles. Wet-dry cycles is a prevalent feature on terrestrial hot springs where life, which is considered as one of the plausible niches where life might have originated. Interestingly, wet-dry cycles have also been shown to promote the formation of peptides from non-activated amino acids like glycine. However, the effect of lipids on this particular process has not been explored yet. Towards this, we looked at the effect of POPC as a model lipid on non-enzymatic glycine oligomerization process under wet-dry cycles. We observed that glycine can form peptides up to hexamers both in the presence and absence of POPC under wet-dry cycle. However, the overall oligomer yield was lower in the presence of POPC. We hypothesized that one of the plausible reasons for this lowering of peptide yield in the presence of POPC could be a cross reaction between glycine and POPC leading to formation of some new product. A preferential utilization of glycine for this cross reaction would render less glycine molecules available for oligomerization, thereby decreasing the overall oligomer yield. Towards this, we systematically checked for the formation of a new product in the glycine plus POPC reaction. Our primary TLC analysis indeed showed the presence of a new band in glycine plus POPC reaction that underwent wet-dry cycling, as you can see in the lane 4 of this TLC image. Notably, this band was absent in other control reactions. Further analysis of the reaction mixture with high resolution mass spectrometry revealed the presence of two new compounds in the reaction, namely N-palmitoyl glycine and N-oleoyl glycine. They belong to a class of amphiphiles called as N-acyl amino acids. As both N-palmitoyl glycine and N-oleoyl glycine have a similar polarity, they appeared as a co-spot on a TLC plate. The identification of N-acyl amino acids in the reaction mixture gave us a clearer picture of what was happening in the glycine plus POPC reaction under wet-dry cycles. There are two competing processes which were simultaneously occurring in the reaction where glycine can react with other glycine molecule leading to formation of peptides, or it can react with POPC to form N-acyl amino acids. Both these reactions depend on the free glycine monomer pool in the reaction mixture. After confirming the formation of N-acyl amino acids from glycine and POPC, we asked whether this reaction is specific to only glycine and POPC or it can be observed with other amino acids and lipids as well. Towards this, we systematically varied the amino acid and the lipid component of the reaction mixture and checked for the formation of N-acyl amino acids. For the amino acid variation experiments, we checked POPC reactions with alanine and valine, where we could observe the formation of N-acyl amino acids corresponding to alanine and valine respectively. For lipid variation experiments, we performed glycine reaction with short chain phospholipids such as C10 phosphocholine and C8 phosphocholine. In these reactions as well, we could detect the formation of short chain N-acyl amino acids like N-dekenyl glycine and N-octanyl glycine. Overall, these results confirm that the formation of N-acyl amino acids was not specific to only glycine and POPC and can be observed with other amino acids and phospholipids as well. The formation of N-acyl amino acids by the reaction between an amino acid and phospholipid can be mechanistically explained as a classic example of an ester-amide exchange phenomenon, where the nucleophilic amino group of an amino acid can attack one of the ester linkages of phospholipid to form an alcohol and an amide, which is N-acyl amino acid in our case. This process occurs more readily at higher temperatures such as used in this study. Interestingly, the ester-amide process has been shown to facilitate the formation of deoxypeptides from amino acids and hydroxy acids. However, such phenomenon has not been reported for amino acids and lipids. Our study is the first experimental demonstration of ester-amide exchange occurring between amino acids and lipids under prebiotically plausible wet dry cycling conditions. This process led to the formation of an interesting class of prebiotically plausible single-chain protoamphiphiles called as N-acyl amino acid. The N-acyl amino acid is a hybrid of a fatty acid and an amino acid which are linked via amide linkage. Given the structural similarity of N-acyl amino acids between fatty acids and N-acyl amino acids, we wonder whether N-acyl amino acids can themselves act as a protocell compartment. Fatty acids, which are widely considered as a constituent of protocell compartment, form vesicles when the pH of the solution is equal to the apparent pK of their head group. At this pH, the protonated and deprotonated species, which are present at fairly equal proportion, come together to form a pseudodiacyl structure, which is stabilized by hydrogen bonding. This, along with hydrophobic interactions, allow fatty acids to form vesicles. The apparent pK of most of the fatty acids is around 7 to 9. Therefore, they can form vesicles only in neutral to alkaline regime. However, the vesicle formation behavior of N-acyl amino acids has not been explored yet. Towards this, we systematically checked their self-assembly behavior using two N-acyl amino acids, namely N-oleoyl glycine and N-oleoyl serine. As you can see from differential interference contrast and fluorescence microscopy images, both these N-acyl amino acids form vesicles in acidic pH range, which is markedly different from fatty acids. The optimum pH range for vesicle formation by N-oleoyl glycine was found to be between pH 5 to 6, whereas that for N-oleoyl serine was between pH 4 to 5. Although both N-acyl amino acids and fatty acids have a terminal carboxyl group in their head group, the presence of amide linkage in N-acyl amino acids might lower the pH optimum for vesicle formation in the case of N-acyl amino acids. Furthermore, the preferential pH for vesicle formation also seems to depend on the nature of the amino acid component of N-acyl amino acid. As you can see here, that the pH optimum of N-oleoyl serine is slightly lower than that of the N-oleoyl glycine, maybe because of the presence of an electron withdrawing hydroxyl group in the side chain of serine. This ability of N-acyl amino acids to form vesicles at acidic pH could have provided them a selective advantage over fatty acids to form protocell compartments under acidic geothermal pool-like conditions. Overall, We showed that when a mixture of glycine and POPC is subjected to wet-dry cycling, two concurrent yet competing reactions occur in the reaction mixture, leading to the formation of peptides and N-acyl amino acids. We further showed that the formation of N-acyl amino acids was not specific to only glycine and POPC and could be observed with other amino acids and phospholipids as well, indicating the generality of this reaction. The N-acyl amino acid synthesis by the reaction between an amino acid and a phospholipid can be mechanistically explained as an example of an esteramide exchange process. N-acyl amino acids are able to form vesicles at acidic pH, suggesting their potential to serve as protocell compartment under acidic geothermal pool-like conditions, which are considered as one of the plausible niches where life might have originated on early Earth. All the work that I have explained in this talk has been recently published in Chemical Science Journal. If you are interested in knowing the finer details of this work, then do check out our paper. Thank you for listening to this talk. If you want to know more about our research interest or have any questions, then please do get in touch. We'll be happy to discuss. Thanks a lot. Have a good day.